that ends up affecting the evolutionary trajectory. So if you think about a bacterial population, and by the way, from this point onwards, whenever I say population size, by and large, I'll be referring to bacterial populations. So in such populations, when you start them from a single clone, then all else being equal, the size of the population determines two things. It determines how much of genetic variation is there in the population, because in such populations the variation comes from the mutations, and more the number of individuals, more the mutations. And it also depends on the efficiency with which selection is working across those mutations. So the efficiency of selection also is known to go up as population size increases. So there is a slight bit of a push and a pull between these two factors, but it's more or less accepted, and people have shown with several experiments that large populations, all else being equal, tend to adapt faster. However, if you think about it, there is a little bit of a possibility of a complication over here, and this is where our thing starts. So remember I said that mutations are important. However, we also know that not all mutations are born equal. Now, of course, mutations are random with respect to fitness, that all of us know. However, if you look at mutations, we know that there are some mutations which are beneficial, but a large number of mutations are actually deleterious. They reduce fitness, and the effect size, so the amount by which these mutations are increasing or reducing fitness, those effect sizes are also different. So in general, people have done a lot of empirical studies to show that getting mutations which are both beneficial and have a large effect size, that is something which is relatively rare. Okay, this is what comes from the shapes of the distributions of fitness effects that people have empirically constructed. Now, if beneficial mutations of large effect sizes are rarer, then it follows that small populations and large populations are going to behave slightly different. So in small populations, the evolution will be primarily driven by mutations with small benefits, because these are the mutations which are more common and therefore they are accessible to the small population. The large populations are going to be driven by mutations with large benefits, because such mutations, although rarer, will actually be accessible to the large populations because of their larger size. And once they come, they will obviously be able to increase the fitness of the larger populations by quite a lot. Now, if this were to be the case, then what will happen after a population has already evolved at a particular size? So if I have two populations which have evolved under the same scenario, and let's assume that even the genetic background to begin with were the same, then after the evolution has happened, and now I subject them to a different environment, am I going to get a difference in terms of how they are going to respond? Now, this in some sense is, if you look at it in one way, obviously, if I'm saying that the mutations are different, then the genetic backgrounds after the evolution has happened is different. And therefore, the trivial answer to that is no, they are not expected to be the same. However, can we find some patterns in that? And that is the stuff where we began our study. However, before we get into that, we need to quickly understand what is meant by the size of a population, a bacterial population, particularly when you are working with it in the lab. So as we know, in a lab, we will typically culture a bacterial population in a flask or some kind of a plate, and there will be some medium in that. And I don't know if you can see this, shaded thing. This is the medium. And to this, you introduce some number of bacteria, the initially occurring, say a number N0. And this bacteria then grows to a final population size of NF. And it takes about, say, G generations to do that. Then, if you think of a simple exponential increase, then it can be very trivially shown that NF is related to, or NF is equal to N0 into 2 to the power G. So now that we have two different sizes, NF and N0, which is a relevant measure of population size? And obviously, you keep on doing this, and this is how the whole thing goes. You take it forward, your experimental evolution in the lab. So now that you have both NF and N0, which is a relevant measure of population size, and turns out that the answer is neither. So it has been shown theoretically that the evolutionarily relevant measure of a population size, when the population numbers are fluctuating in the way that I talked about, the evolutionarily relevant measure is the harmonic mean, which is N0 into G. Now, at some other place, we have actually vigorously argued against this particular measure, but that's a story that doesn't affect the current story in any particular way. So for this present purpose, we are going to assume that this is the evolutionarily relevant population size, which means that the higher the N0 into G, you expect the population to evolve faster. You expect them to hit the fixation events, a mutation in them to become fixed much faster. So, in order to answer our question, we did some experimental evolution using E. coli. So we started our experiments from a single colony of E. coli NG1655, and we had two different measures of HM, harmonic mean, and these were attained in two different ways. So in one case, we started with a small value of N0, and then we allowed it to go for a longer time, and then we subcultured it for the next generation. So this is the one that gave rise to a smaller N0, a smaller harmonic mean. And we had another treatment in which we had a larger N0. So basically, the bottleneck ratio was larger, 1 in 10 bottleneck ratio. And this one was 1 in 10 to the power 4. And then we allowed it to grow for a smaller amount of time, and then we subcultured it back to this N0. So just to tell you what this led to, it led to three different treatments. So in these, we call them SS, SL, and LL. And in this, the first letter gives you a relative measure of the harmonic mean, which is either small or large. And the second gives you a relative measure of the NF, or the culture volume, which was again small and large. So for each type, we had 8 independently evolving replicates. And the harmonic means, the large harmonic mean was of the order of 10 to the power 10, and the small harmonic mean was of the order of 10 to the power 6. So you can see that this is small only in relationship to this. This is still by itself a pretty large number. So we took these 8 replicate populations, and now we needed an environment in which to evolve them. So we took an environment in which we had a sublethal concentration of 3 different antibiotics, norfloxacin, rifampicin, and streptomycin. And as you can see, that they work very differently. So they are from very different classes. And in terms of fitness, we measured 2 things: the carrying capacity, which is the maximum OD reached during population growth, and the growth rate, which is the maximum slope over moving windows of 10 reading. So we first evolved these populations under these conditions for about 380 generations. So here I am showing you fitness in terms of carrying capacity K. This is fitness in terms of growth rate R. Everything has been normalized by the ancestor's value, which is why the starting point is 1 for everything. And the error bars are standard error. So if you look at these two graphs, you see 2 things. First, the black one, which is the large effective population size, they evolved much faster compared to the other things, which is, as I said, not unexpected. And secondly, but more importantly, after 380 generations, the fitnesses of these things, that is at this point and this point, the fitnesses of these things were not statistically different. So they were statistically indistinguishable from each other at this point. So now they have evolved. So again, just to give you a schematic of the whole thing. So we have 2 populations, or 2 sizes, large size and small size. These have evolved faster, these have evolved slower. After 380 generations, they have reached the same point. And now we want to see how they will do if we put them in a different environment. And in order to do this, we measured 2 different things. We measured what is known as the efflux activity, and we directly measured their fitness in novel environments. So why efflux activity? Prior to that, what is efflux activity? There might be a few people who are not bacterial evolutionary biologists, so I'll quickly mention what is efflux. So basically, whenever there is something inside a bacteria which it doesn't like, one of the mechanisms by which it takes care of it is by simply throwing it out to these so-called efflux pumps, which are basically membrane-bound proteins. So these proteins, they use up energy from ATP, and they throw out all kinds of things, including antibiotics, heavy metals, salts, mutagens, etc., etc. So this is known to be one of the major mechanisms by which bacteria fight a broad range of stresses. And our own work and other people's work had also shown that if efflux activity goes down, then the ability of the bacteria to face a large number of things, a large number of stresses also goes down. However, in this particular case, remember our selection environment is a cocktail of 3 different antibiotics. And antibiotics are one of the biggest class of things that efflux pumps throw out. Therefore, there is no reason for us to expect that the that the efflux levels will go down. However, to our utter surprise, we found that in the LL environment, in the LL treatments, the efflux activities of 7 out of the 8 replicates went down significantly. In one case, it was the same as the ancestor. So the ancestor is this dotted line. In the small populations, the efflux activity went up. Okay. But this is a very strange thing, because in an environment where there is antibiotics, you don't really expect this stuff to happen. Anyway, we wanted to check what is happening in terms of other environments. So then we, as I said, we tested, since our own studies had shown that efflux going down affects their fitness in other environments, we tested the fitness in 4 other novel environments. So we chose one antibiotic, ampicillin, which acts in a way which is very different from the other 3 antibiotics. We chose one heavy metal, copper, and we chose 2 carbon sources, sorbitol and urea. And to our utter surprise, we find that the large populations, which are expected to have a greater variation, they actually have lower fitness in terms of K in all these 4 environments. And in terms of R, they have lower fitness in 2 of the environments, and in the other 2, their fitness is not significantly great. So overall, one can say that the LL populations are doing worse in the alternative environment. Now, here one has to note that the reduction in fitness in ampicillin and copper can be directly related to efflux. However, it's not intuitively obvious how the fitness reduction in sorbitol and urea are related to reduced efflux activity. So, what we saw was that adapting slowly in smaller numbers are making asexual populations less vulnerable to subsequent environmental changes. Now, why is this reduction happening in the LL population? Now, whenever you are working with population size, the first thing that comes to your mind is drift. Maybe the LL population is facing drift, but then drift at population sizes of 10 to the power 10, that doesn't sound right. More critically, there are 3 other observations which suggest that drift is probably not playing a role over here. Why? Because as I said, if anything, you expect the SL and SS, the ones which are having a higher, lower population size, to face greater drift. But these are the guys who are actually not losing the trait. These are the guys who are losing the trait. Secondly, remember there was one guy over here among the SLs which had a lower efflux activity just like the LLs. So turns out, if you look at the speed of adaptation of this particular replicate actually adapted much faster than all these other guys. As a matter of fact, it adapted at a speed which was reminiscent of that of the LLs. Now, this is not the kind of behavior that you expect when you are getting drift and mutation accumulation through drift. More critically, if you have drift leading to all these things, then you expect the variation across your replicates to go up. But if you just look at it, you can see that the variation across the replicates is not really different. We formally tested it, and it was statistically similar, no significant difference in terms of the variation. And finally, population genetics calculations tell us that 400 generations is too less to see parallel increase in a trait just by mutation accumulation and non-selection. So taken together, our contention is that it is selection which is leading to this result and not mutation accumulation through drift. Now, if it is selection, then this is becoming even more interesting. Why? Because think about it. We all know that a biological character can, if it is not essential or if it is disadvantageous or costly, then it can go down in a population. We have any number of examples of that in the biological literature. However, when that happens, you expect that the trait in question has gone down because the environment in which the evolution was working, in that environment, that trait was bad. However, that is not the case over here. What we are having over here is the same environment, the same starting population. So the genetics, the genetic background right at the start is also the same. And yet, when they're evolving, you are actually getting completely different evolutionary outcomes based on the population size. So how on earth is this happening? So to understand why this is happening, we essentially needed to make two key speculations. One speculation was that, it's known that efflux activity gives you higher fitness when you are facing antibiotics, particularly when you are facing multiple antibiotics. Efflux is very, very important. However, we also know that efflux is energetically costly. So one possibility is that the LL populations, they were able to arrive at rare mutations, and the mutations have to be rare, which is why only LL could arrive at them and SL and SS could not. And once they arrived at those mutations, then the LL populations no longer required the costly efflux activity to fight the antibiotic. So at that point, the LL could actually afford to lose all the costly efflux activity, whereas the SL and the SS, they did not have this option, as a result of which they had to stick with higher efflux activity. So this is a speculation. Can we really check this out? Now, checking this out in terms of an experiment, I won't say it's impossible, but it's very, very tough. And I'll discuss why it is tough in a few minutes. So what we did instead was we went for some very simple, overtly simplistic, almost toy model kind of simulations to figure out what are the minimal requirements for this kind of a divergent character evolution when they're in the same environment starting from the same genetic background. Now, what we did some Wright-Fisher simulations. Now, our model requires that whatever trait we are talking about, that is going to be bad in the larger population, but beneficial in the smaller population. And if you think about how the mapping happens between the expression of our focal character and fitness, there are two ways in which this can happen. One, of course, is the environment is different. And the other is that the genetic background on which these mutations are operating, in some way, even though they start from the same source, but in some way the genetic background leads to a difference in terms of how the mutations are going to work. So the first one is obviously not the case because, as I said, experimentally we made sure that the environment was the same. But then there is a possibility about the second one. And the second one will work if and only if we have sign epistasis, which means that the same mutation is going to have a positive fitness effect in one genetic background and a negative fitness effect in another genetic background. So to study this, we came up with a very, very simple two locus T allele model. So our model has two traits, call them trait A and trait B. So trait A is the one that we are going to focus on. So trait A in the context of our experiment is analogous of efflux. And trait B is something that also affects fitness under those conditions, but it has a lower mutation rate compared to the trait A. And for each trait, we said that let there be three states, intermediate value, which is O, and a high value H and a low value L. Why did you do that? Because that's again, reminiscent of our experiments. We had the ancestors, and then some guys went up, some guys went down. And note, although here I'm calling them as traits and trait values, you can also think of them in terms of loci and alleles. You are going to reach roughly the same conclusions. So we then said that for this whole thing to work, the way in which we think about mutations has to be very different, because the whole idea was that efflux kind of mutations are easier for everybody to reach, which is why the small populations can reach them. The other antibiotic binding mutations are something which are harder for them to reach, and therefore only the large populations can reach them. So, and we also know that the mutations which increase fitness are less common. Mutations which decrease fitness are more common. So here we have, let's say, A, and this is both 0 to show ancestor and O to give you the intermediate one. So this, from here going to a lower trait state, is very common, 10 to the power minus 3. And going from here to the opposite side is rare. Similarly, going from AO to AH is the mutation rate is lower, 5 into 10 to the power minus 5, but going from here to here is higher. Now, how did we arrive at these various mutations? Now think about it. What is the lowest mutation probability in a genome? The lowest mutation probability in the genome is going to be whatever is the per base pair mutation rate under normal conditions. I mean, we are just talking normal conditions here. So whatever is the per base pair mutation rate, that is going to be the lowest that you can go. And that, in the context of E. coli, is about 10 to the power minus 9. What is the highest mutation rate, again under normal conditions, that one can get? The highest mutation rate is going to be roughly equal to the per genome mutation rate per generation. And in the context of E. coli, again, that is somewhere about 10 to the power minus 3. So in some sense, these are very rough, thumb rule kind of things, but in some sense, these are the two boundaries, 10 to the power minus 3 and 10 to the power minus 9. And then all these other rates, we simply put them somewhere in the middle, 10 to the power minus 5, 10 to the power minus 4, minus 8, etc., just to have some mathematical or whatever, some probabilities assigned to our values of common, into our intuitive notions of common, very common, and so on. So these are the rates that we set for locus A. And what we said was that the mutation rate at this locus are higher compared to the mutation rates over here for the other locus. So what we are talking about is a differential mutation supply across traits A and B. Now, in order to take care of the sign epistasis, what we did was we had a very simple 3 into 3 9 state thing like this. So here, if you look at it, if the trait value of D increases, so if you go from left to right, the fitness values always increase. So, 5, 6, 7, 3, 4, 8, 1, 2, and 9, they always increase. However, in the context of A, the trait values, when you go from L to H, increases if you have a BL or a BO background. However, if you have a BH background, it goes in the reverse direction. So this is what we mean by sign epistasis. So all the simulations started from this guy over here, whose fitness, which is the OO, the intermediate stage in terms of both characteristics. And what we did was we simulated them for 1,000 generations. And after 1,000 generations, we figured out what is the composition, the genotypic composition of the population. And for each one of these, we had 10 replicates. So what we are showing you over here is time on x-axis and the fitness on the y-axis, and what is the the time trajectory of fitness in the population. So as you can see, when the population size is low, about 10 to the power 3, then the fitness just goes to somewhere about 6. When it is 10 to the power 4, the population size is 10 to the power 4, it goes much higher. And when it is 10 to the power 6, it very fast reaches the highest point, which is about fitness of 9, and everybody then stays there. So larger populations are adapting faster, larger populations are adapting better. This is what we saw in our experiment. Cool. But now, what we looked at is what is happening to the character states of A and B. So here, wherever you have a green arrow facing this side, right side, it's a high efflux state. Wherever you have a red arrow facing left side, it's a low efflux state. And this one is the wild type. So what you can see is averaged over 10 replicates. When the population size is population size is low, all these populations are settling down to high efflux. However, as you keep on increasing the population size, so this is 10 to the power 2, 10 to the power 3, all the way up to 10 to the power 7, as you increase the population size, the fraction of this one, which is the low efflux thing, that keeps on going up. And by the time you have hit 10 to the power 5, most of them are here, which is low efflux, a few are here. And by the time you hit 10 to the power 6 and 10 to the power 7, everybody actually has hit low efflux. Now, this can happen in two ways. One is you have a polymorphism in the population, or the other is that out of the 10 replicates, some of them are going here and some of them are going here. Some of them are going to say high efflux and low efflux, and some are going to high efflux. So to figure out what is happening, we actually looked at the genotypic composition of all the replicates after 1000 generations. So here, these white boxes, they are simply indicating presence. Okay, and the black box indicates absence. So here on the x-axis, you have the 10 replicates, and on y-axis, you have the 9 genotypes. So what you can straight away see is that for every single replicate, it's one genotype and nothing else. So there is no polymorphism happening over here. It is simply what fraction of the population is, or what fraction of the replicates is going towards high efflux versus low efflux. So what we ended up seeing over here is that as you increase the population size, you are going to have low efflux evolving in these kind of conditions when you have sign epistasis and when you have mutations, differential mutation rates acting together. Now, obviously, you need the controls for this. So what will be the control? First, you remove the sign epistasis. So this is what was done. So remember, in the other case, it was 7, 8, 9 in this direction. Now we reverse this fitness landscape. The moment you do that, so now it's all in the same direction. The moment you do that, everything just goes towards high efflux. So you don't have low efflux coming at low population size. You remove the differential mutation rate across the two loci in whatever way you remove, and we tried out multiple ways. We find that irrespective of population size, you end up going into one state or the other. So basically, this is one combination of mutation rates in which everybody evolved low efflux. There are other ways of other values of mutation rates for which everybody will evolve the high efflux. So the conclusion from this was that sign epistasis and differential mutation supply, both of them are together at the minimum requirements for the kind of divergent character evolution that we saw, if it has to happen in the same environment. However, there was still a problem. And the problem was in terms of the first fitness graph that I showed you from the simulation. What is it? So if you look at it, this is showing that when the population size is 10 to the power 3, the overall fitness is going to be lower. When the population size is higher, the overall fitness is going to be much higher, which means that after 1000 generations, they are going to two different fitness levels. However, if you remember our experiments, after 380 generations, both the small and the large populations had reached the same fitness level. Now, if that were to be the case, how does one get that from these kind of simulations? And turns out that if you are simply doing a two locus kind of a situation, then whatever mutation rate you do, whatever, as long as the fitness landscape is simple of the kind that we did, you just cannot replicate this particular result. So the reason this is so is because it's pretty obvious that the large populations are taking a different mutational route. The small populations are taking a different mutational route. So if that were to be the case, then we are essentially talking about two different optima in the fitness landscape. And that two different optima, it is impossible to get that in the kind of simple fitness landscapes that we were doing, obeying the sign epistasis constant. So in order to do that, we did a second order modeling, a different model, in which it was a 3-trait system. And in this, we added a trait called C. And this C, you can think of it as like an efflux pump, another efflux pump. So it, for all practical purposes, behaves the same way as A. And It has the same mutational properties as A, but both A and C have a sign epistasis with B. So this is what the fitness landscape looked like. So the main point to note over here is that in this particular case, we had two different global optima. I mean, the global optima at two different places, which can be reached through two different mutational routes. And these were the ones that had a value of 14. So long story short, if you now simulate this kind of a model, then this kind of model will actually lead to a situation where the fitness can be both, fitness can be high in both the small populations and the large populations. And at the same time, you can get back that observation that the large populations are going to have, the small populations are going to have high efflux and the large populations are going to have a lower efflux. In other words, this qualitative change is getting captured over here. So at least technically, these kind of two sign epistatic traits can potentially explain the evolution of efflux in our experiment. Now, please note that this is just a simulation. Okay, the bacteria are under no obligations to follow these assumptions, and the actual case can be far, far, far complicated. So, for example, we can still have it two traits, but then the fitness landscape between them can be way more complicated than the simple 3 into 3 9 state that we took. It can be non-monotonic and all kinds of stuff. There can be many more dimensions to this landscape compared to the simple 3 that we took. So obviously, the way to figure out which one is happening is that you do a whole genome sequencing, you figure out which are the mutations which are different, and then you fish out those mutations, you put them in a different genetic background, you show where the epistasis is happening, and so on and so forth. Doable. There are people who are doing it. We are not in a position to do it, and frankly speaking, that's not what interests us either. So we did not go any further along this line of investigation. We took up the other line, which was the point that the larger populations are going down in fitness. Now, please observe that in the context of the efflux, we had to posit somewhat complicated genetics to see our results. Now, is that also the case for the other part? Is it the case that this kind of large populations using fitness in other environments is going to happen only for very few kinds of environments, for example, the kind of antibiotics that we took, and not for something? Therefore, and the other thing that we were also interested in this context is how fluctuating environments end up affecting this relationship. And the reason fluctuating environment is because we have been working on fluctuating environment for the last few years. So what we were really interested in is the trade-off or fitness cost of evolving under a large or a small population. Now, I appreciate over here that trade-offs and fitness costs have been defined in many, many ways in the literature, and 20 people probably will see it in 200 different ways. So I'll be very strict in terms of defining what I mean by trade-offs or fitness costs over here. So what I mean is that when there is a statistically significant fitness increase in one environment and a statistically significant fitness decline in another. So only when these two things happen simultaneously will I call it a trade-off happening. Now, if you look at the literature, such trade-offs are widely expected. However, when one looks at the experimental literature, particularly in the context of microbes, they are not that widely found. And there are several reasons that have been given to explain why they are not as common as people expect them to be. So lots of people have said maybe they are not there at all. And for example, a recent paper has shown that only 11% of some 80 mutations that happened in a context exhibited antagonistic neutron. The other people say that the reason is more prosaic. The trade-off is very much there. However, you need to check the fitness in appropriate environmental conditions, and maybe the environmental conditions that you are checking them under are not those, so you're not checking them properly. The second point is that, remember, if I go by this definition, this is a very stringent statistical requirement. So maybe the experiments that lacking the statistical power to see, one statistically significant thing in one direction and another statistically significant thing in another direction for something that has evolved in the same environment. And another, thing that people often state is that, trade-offs will take some amount of time to evolve. So if you are having a Lenski kind of an experiment, worth thousands and thousands of generations, yeah, sure, that's where you expect trade-off. But if you are doing something over, let's say, a few hundreds or a few thousands of generations, then probably your experiment is not sufficiently long for you to see trade-offs. Fair enough. Another proposed reason that came up recently, and this is the reason for which fluctuating environments was of interest to us, was that if the difference is in terms of whether the environment is homogeneous or heterogeneous. So if you have a population which is evolving in a homogeneous environment, then all the mutations which are evolving in response to that environment, they are blind to the fitness effects in other environments. Therefore, when you suddenly put them in other environments, selection sees them and therefore the fitness goes down. However, if you have a heterogeneous environment in which the environment is changing very often, then whenever there is a change or whenever there is a mutation in one environment, soon after it experiences the other environment and therefore selection is no longer blind. So at this point, you don't really expect after a long time costs to evolve, simply because during the selection process itself, whenever the environment has changed, things have been taken care of. So, in other words, selection in heterogeneous environments are expected to lead to avoidance of fitness trade-offs. Now, this particular reasoning did not entirely sound convincing to us, because when we were looking at heterogeneous environments, we could see a few studies which were detecting trade-offs. So what we did was a very simple exercise. We simply took all those studies, in heterogeneous environments which were showing trade-offs, and then we arranged them by population size. Why population size? Because we were by then convinced that population size is a big player in this whole. And when we did that, so these are some 8 or 9 studies that we could find in the literature in which we found that people have explicitly looked for cost. Whether they're found or not is a different thing. And we could actually figure out what the corresponding population sizes were. And when we plot them in ascending order of population size, we find that all the studies which are finding costs are the ones which have evolved at relatively small population sizes. All that which have evolved at relatively larger population sizes are not finding any costs. Now, obviously, this was exciting for us. But then, if you go around finding for pattern, you find all kinds of patterns at all kinds of places. So obviously, one has to go beyond this. So what did we do? I mean, again, we did experimental evolution, again, E. coli, again, MG1655. So this time what we did was we had 5 different treatments. So in one of these treatments, the carbon source was fluctuating every 13.3 generations, and it was fluctuating over 4 different carbon sources: galactose, maltose, sorbitol, and thymidine. And the corresponding controls were treatments in which these carbon sources were continuously present. So there are no fluctuations happening. And for each one of these 5 treatments, we had a large population size with about 10 to the power 8 harmonic mean, and a small population size with about 10 to the power 5 harmonic mean. So 6 replicates per treatment, 60 populations in total. We did this for about 480 generations, and then we measured their fitness in all these media, or all these 4 media. So about 240 assays. So this is a slightly busy graph, but I'll try to walk you through this. So here we have 4 panels. So in each panel, what we have is evolved in a particular homogeneous environment. So evolved in thymidine, galactose, sorbitol, and maltose. So you have 2 kinds of arrows. So your fitness on y-axis, and the assay environments, galactose, maltose, sorbitol, and thymidine, are on the y-axis or on the x-axis. And you have 2 kinds of arrows. The red arrows are points where you are seeing costs in both large and small environment. And please remember, in our case, cost is when the environment in which they have evolved has a significantly high fitness compared to the ancestor, and the other environment has a lower one. So in this case, in both the small and the large populations, you have a fitness decline in the other three environments, whereas the environment in which you have evolved, thymidine, the fitness has gone up significantly. In the case of galactose, when they have evolved in galactose, the fitness has gone up, but only for the large population, the fitness has actually gone down in this case when they are acid in thymidine. In the other cases, you actually don't see any cost because either they have failed to evolve in their own home medium, or they have not paid, they have not significantly reduced fitness in the other one. Oh, sorry, I forgot to tell you that these bands are the 95% confidence interval bands. So just to give you the nutshell, both the large and the small populations evolved costs in thymidine. However, the larger populations paid greater cost, which means that the decline in them was larger. Only the large populations evolved in galactose paid cost in thymidine. No population, large or small, could adapt without paying costs. Now, if you look at what was happening in the fluctuating environment, you see something completely different. Here, the fluctuating large adapted simultaneously to two environments, galactose and thymidine, and this one, galactose and thymidine, and avoided the cost of adaptation in the other. Okay, so you have increase in fitness in two, avoidance in the others. However, if you look at the small population, which is this blue one, you can see that although it evolved in thymidine, so it evolved greater fitness in thymidine, it actually paid a cost in the other. And these are all significant. Okay, now please remember, these are populations, the F treatments, in which both the large and the small populations have seen all the carbon sources. Okay, in spite of that, they are able to evolve in T and G, no loss of fitness in the other two for the large, and for the small one, they evolve higher fitness in T, but loss of fitness here. Okay, Manuel, I'm slightly running out of time. I'll finish before 1 hour, but I'll need about 5-7 minutes more.
That should be fine.
Thank you. Okay. Now, obviously, when you're talking about a fluctuating environment, the relevant measure of fitness is not the arithmetical mean of the fitnesses, but the geometric mean. So we computed the geometric mean, and not surprisingly, the geometric mean of the large populations was significantly larger than the ancestors. Very interestingly, the geometric mean of the small fluctuating populations was not significantly different from the ancestors. So in a nutshell, what are we seeing? We are seeing that in homogeneous environments, the larger populations are evolving greater cost, but in heterogeneous environments, the larger populations are avoiding the costs altogether, whereas it's the smaller populations which are evolving costs. So when you have large size, population size, and environmental heterogeneity present together, only then you can ensure a simultaneous adaptation without paying a fitness cost. This is not what was hypothesized by these other people whose quotation I showed you. But then, obvious question is, how on earth is this possible? Because as I argued, when you have a heterogeneous environment, then selection is not blind to the pleiotropic effect. So if it is not blind, then how can FL populations become simultaneously better at multiple environments without paying any costs anywhere? So the way we explained it was that large populations, as I said, there are two opposing forces. One force is that due to larger size, more mutation, more variation. And the other force is that due to higher selection, less variation. Now, the possibility is that in this context, the first effect, this more mutation effect, is actually stronger than the second effect. Okay, so how does one test that out? So sorry, the first effect is more stronger than the second effect, due to which you have secondary compensatory mutations that are coming out in the large populations, which are able to compensate for the antagonistic pleiotropic effects that you see in the small population. So how does one check this out? In order to see whether what we are saying is right or wrong, we did a whole genome, whole population sequencing of 3 replicates each from FL and FS. And this is the nutshell. So I'll quickly walk you through this. So here on top, you have the 3 large populations. On bottom, you have the 3 small populations. Wherever you see a label which has a brown thing, a brown background, so that those are the ones in which the galactose utilization genes have a mutation. And wherever you have a blue one, those are the ones where you have a thymidine utilizing mutation. The first thing that you see is that the number of mutations that have been enriched, and I mean more than 10%, in the large populations is much larger compared to what has been enriched in the small population, which means these guys are actually having more number of mutations. Second thing is, if you look at the identity of the mutations, what are these mutations doing? You find that in the small ones, you only have mutations. I mean, I'm talking about the known ones. You only have known mutations for thymidine utilization or uptake. However, in this one, in the large ones, you have both thymidine, the blue ones, and galactose utilizing mutation, which means that the antagonistic pleiotropic effects of thymidine related mutations are probably getting nullified by these galactose utilizing mutations. Note that none of them are here. And one interesting thing, this is of interest to population geneticists, note that here in both the small and the large populations, we are actually seeing multiple mutations fixing. Now, this is very interesting because remember, this is just a 480 generation long experiment, and this is fluctuating environment. So, wisdom, population genetics wisdom tells us that you don't really expect fixation events to happen so fast. That also in fluctuating, and that also multiple ones. Okay, so we were very tickled to see this particular result. Anyway, now the question is, what is the way in which the cost is getting avoided? Now, based on that mutation result that I just now showed you, the argument that I'm trying to make is that probably the individuals have both T and G utilizing mutation. That's one possibility. But the other possibility also is that you have subdivision into specialist subpopulations. So half the population or whatever, some fraction of the population will utilize T, the other fraction will utilize G, and thereby they will be able to be together. Each one will be able to compensate for the other. So in order to figure out which is the case, what we did was, since we had the mutation frequencies, we actually computed the probabilities of one individual carrying both T and G mutations in FL. And we actually found, so these are for the 3 replicates, that in all 3 replicates, the probability that in all 3 replicates, more than 50% of the individuals are expected to carry both mutations simultaneously. So which suggests that probably based on this data, we can say that here we don't have subpopulation evolution. Here, each individual has actually been able to evolve the ability to take care of both kinds of targets. So just to summarize, so what we showed was that even when you start from the same genetic background and work under the same environment, different population sizes can change the fate of traits. In other words, you can have gain of a trait or a loss of a trait. And the genetics of this phenomena is likely to be complicated, but should depend on sign epistasis and differential mutation rates for the 2 traits in question. When you are evolving under homogeneous conditions, it can lead to fitness costs in other environments. And when you have fluctuating environment, then things will change. And now larger populations can end up avoiding this cost by acquiring more mutations in the same background, which means it's the smaller populations which will end up paying the greater cost. Now, this is for scientists. Now, you know that we also need to get grants. So what are you going to tell a grant manager? Let's. So the non-technical implications of what we have done is that natural populations, we know they typically inhabit fluctuating environments. Now, obviously, depending on their ecology, different bacteria will have different population sizes in the environment. Now, if you look at our result, what we are saying is that under fluctuation, whether a population is able to evolve cost-free or not is a function of its population size. And therefore, the combination of our insights with information about bacterial ecology and particularly about what kind of population sizes they find themselves in nature is going to lead to insights about which bacteria are likely to evolve multi-drug resistance or other properties like that, and which bacteria are not expected to evolve that. So this is the penultimate slide. So this stuff that I presented is the thesis work of Yashraj Chavan. Frankly speaking, he knows a lot about this than I do. So if you have very tough questions for me, I will probably just end up passing them to Yashraj. Shraddha was a PhD student with me, and she actually helped in the efflux related assays. Saumil, he was an undergrad at ICER Pune, and he was the one who worked with Yashraj on those cute simulations. So he actually did a lot of really fancy computational jugglery to make sure that the simulations happen with whatever things that we have. There are two more people who contributed to this line of work, but unfortunately, the precise stuff that they did, I could not show them today. So one is Sarthak, who is at Montpellier. So Sarthak was the one who was responsible for extending this work and trying to show us how this leads to niche expansion. And Irfan was the person who actually simulated the effects of having different population sizes and what that will do to the environmental trajectory. And of course, there is Department of Biotechnology who funded us. And yeah, that's about it. Thank you very much. I'm really sorry, I am slightly over time, but I hope I can take a few questions.
Thank you so much, Sutirth. Yeah, we can have a couple of more questions even after 12:30. So don't hesitate to post your questions to the Q&A tab. I'll start with Sébastien Pochmaille, who should be able to talk right now. Sébastien, if you want to switch on your microphone.
No, I can see the question.
Yeah, so Sébastien, you can also directly interact with Sutirth.
Yeah. Sébastien, is this the question, or you want to ask something more?
No, that's the question. Yeah.
So what Sébastien is asking is, do you always need uneven mutation rates between traits to explain your data? So we actually tried equalizing the mutation rates, and it didn't work. It simply didn't work for us. If you have traits A, B, C with same rates, does that not work? No, it doesn't. On one side, you have two traits versus one on the other side. Hence, this could be seen as twice as many changes. Yeah, absolutely. Yes, absolutely. What you're saying logically is correct. But when we did the simulations, it didn't work out for us.
If you were to try 10, 15 traits, maybe that's a bit much. Do you think it's because the ratio 1 to 2 is not large enough?
Yeah. See, as I said, if we complicate this, there will be many other ways in which it will come up. There's no doubt about that. Okay. We were in some sense trying to see what is the minimal condition at which we can get our results. I mean, that way, if we complicate our fitness landscape sufficiently with just two traits, but make it very rugged and all kinds of non-monotonicities thrown in, then maybe even with two, we might end up getting there. Okay. But yeah, your general point is valid. If we have more dimensions, then all kinds of things can happen.
I guess what I'm thinking is not necessarily more traits, is that traits that are influenced by more genes. So let's say the trait can be influenced on one side by one single gene versus 20 genes on the other side.
Okay. See, that is why if you look at that, I said that in my head, I consider that thing as a trait. Okay. So it doesn't really matter how many genes are there for my way of thinking. Yeah, in the second model, we did reduce that particular constraint. And see, the point is the way we have done our simulation in that framework, if I increase the number of quote unquote genes, wait a second, what do you mean by genes? Trait states, or do you mean ABC, those ones?
What I mean is, but it's just the mutation rate. In fact, as you said, is not necessarily mutation rate as we understand classically a mutation, but rather mutation in a trait, which might be by different genes. So I guess, yeah, that answers completely the question.
Right. So there lies the probability. Yeah.
Perfect. Thank you.
Great, thank you.
Michael?
So there is a question by Michael Finnegan, and you should be able to activate your microphone right now.
Hi, can you hear me?
Yes.
Yeah, I can hear you, Michael.
Great. So in your first experiment, the large and small populations evolved the same fitness advantage in that 3 antibiotic condition. Given the fact that the large populations should have access to rare mutations with large effect size, why didn't these large populations evolve a higher fitness? Do you think it's because both populations are near the fitness peak in this environment? You were saying that the efflux pumps can be costly. So why didn't these large populations find mutations that would give a higher fitness advantage without that cost of the efflux pump?
Right. So if you look at it, the higher populations did evolve something to reduce the cost of the efflux pump, right? That's why their efflux went down.
Yeah.
Now, what you're really asking is that why didn't they make it even better?
Yes.
Right? Yeah. So I think that also has to do with the way in which we did the experiment. So in our case, this is not a deteriorating environment. We have these 3 antibiotics, the cocktail of 3 antibiotics, and that's it. Then it becomes a static environment after that. So typically, the thing that you are talking about, that would probably happen if you increase the antibiotic concentrations regularly. Does that make sense?
Yeah.
So that's one part of the answer.
Yeah.
The second part of the answer, as I said, is, again, it might be a time thing. Maybe if we keep it much longer, then we will end up getting another mutation which will take it to the next step. That's another possibility. I can't really rule that out. And the third thing, as I said, they did go down on efflux, which means that they did come up with something else to take care of.
Yeah. It's just the fact that they did come up with something else. I'm just surprised that that other thing wasn't much more beneficial.
But if you look at it, they evolved pretty fast, right?
Yeah,
and then
that's
they
true.
stabilized. What you're really asking is that once they have stabilized, why didn't they go the next step?
Yes, yes.
Isn't it? And there are 2 answers to that. One is, as I said, the selection pressure may not be adequate for them to do that. And the second thing is, this is only 480 generations, right?
Yeah.
So an interaction of the 2 can explain.
Thank you.
Okay, thank you very much. There's a question by Ophélie Rons. Ophélie, your microphone is on. You can ask your question.
Okay, thank you, Sutirth.
Ah.
I had one question about your model was predicting that either you lose net efflux or you keep it, but you're never polymorphic. And
Yes.
what you see as you see a change in the frequency of replicate population in the simulations that have lost it or kept it. And I was wondering whether you could actually compare that to your experimental findings and whether it was not contradictory with the observation that you found that the variance between replicates in, I don't remember which trait, was not dependent on the size of the population. So is it not a source of discrepancy between your model and your experimental results?
No. Because, I mean, 2 or 3 things. I assume that this is the graph that you're talking about, right? In which we ended up finding that everybody is either one thing or the other. There is no polymorphism after 1000 generations. Is that correct?
Yeah.
Now think about this. This is something that we are looking at the equilibrium, right? Now, when it comes to our experimental populations, we don't know where the equilibrium is. So it is entirely possible that we are catching them much before the equilibrium. That's something that
but
is
I
always
was okay. But you have no
always happen.
Yeah, you have no evidence that you see a pattern like this of really divergent trajectories among replicates depending on population size.
See, here we are going to get that if I do show you the graph at say 400 generations or 500 generations. So it's only because we have run it for such a long time does it disappear.
Okay.
Number one. And second thing is, even with the second model, the more complicated model, there you can get all kinds of things. That there it's a slightly more complex situation happening. Here, this is an artifact of the fact that we are using a very, very simple, 2 locus, 9 state fitness landscape.
And I had another question actually about the adaptation to heterogeneous environments. So maybe I'm missed something, but I was not sure how your results were actually, whether they were contradictory or explaining the finding that the meta-analysis that you showed about experiment that I found, cost or not. Well, this one. And also, if you wanted to go beyond bacteria, I mean, in the literature, there's already meta-analysis of local adaptation looking and showing that population size is generally what explains the fact that you detect local adaptation or not. And I was just wondering about whether there's any connection between your findings and that, and whether it's contradictory or the same thing.
See, the problem is that is why I started right at the beginning by saying that when I say population size, I mean bacterial population. Here, the only source of mutation is de novo mutation and nothing else. Only source of variation is de novo mutation. Whereas if you are talking about a sexual population, then a lot of the variation is just going to come from recombinational variation, right? And there, the whole dynamics of standing genetic variation is going to operate very, very differently. So that is why I will not necessarily be willing to extrapolate these results to, let's say, the larger animals that you are talking about, most of which are probably going to be sexual.
Okay.
So does that answer your question?
And what about the meta-analysis? The purple and green curves you showed? Is it consistent with what you find, or is it? I don't.
These ones.
No, the meta-analysis, not the mutations.
Okay. This one. Yeah, this one.
This
right?
one. Yeah.
Yeah. So this one is. See, the problem is that here they are using different kinds of organisms. Okay. You have all of them are bacteria, fair enough, but you have Serratia, you have E. coli, you have Pseudomonas. I don't know what else you have. You probably even have Chlamydomonas might be the case. I'm not sure. So, of course, the ecology is going to be slightly different, but roughly speaking, what you find over here is that the switch is happening somewhere around 10 to the power, between 10 to the power 7 and well below 10 to the power 8. So somewhere around 10 to the power 7 is your switch. Okay. Now, if you look at our thing, you see that one of our HMs is on the other side, 10 to the power 8. The other harmonic mean is on this side. Okay. So in our case, our small population is somewhere here and our large population is somewhere here. So I don't think our results are contradicting this. Our results are, very much fitting within this pattern that we got.
Okay. Thank you.
Thank you very much, Sutirth. So I think we're already quite over time, so we will call this a day. I have 2 small announcements for everyone. So please keep in mind that we will have the Conférence Hospitalaire on the 2nd of July by Hannah Coco, and that we now have a 2-week break because that corresponds to the original holiday plans, so that we will be back on May 7th at 4 p.m. Not at 11:30, at 4 p.m. because we have a guest from the US. So with this, Sutirth, again, thank you very much for this fascinating talk, and have a nice weekend, everyone.
Thank you.